Now moving towards the lab. First thing I do whenever I enter the lab, I enter the lab around 9 a.m. The first thing I have to do is to maintain the temperature log sheets, which is of two types: the room temperature and the fridge temperature. Fridge temperature is more necessary because it maintains the cold chain for the vaccine, reagent kits, etc. And the second step is to cleaning of the lab. I disinfect all the infection and ready the lab for the use. And now I'm moving towards the recruitment side. As I'm working as a lab manager, I have to maintain all the SOPs and all type of recruitment companies which are giving us the test. And I do the files as there is a connectivity issue. And now I am making the attendance for the many of the people around there. I have to maintain their attendance too. All this is basically the static lab, and I have to maintain all the things. I have to work as a multitasker. I am maintaining all type of inventory, and now so I need a tea break. After that I have to move towards the lab, in which I receive a switch on the CBC analyzer, which is a BC2800, and now it is initializing, and I have to wait until the machine is ready. Now I have to rotate the CBC sample, which is an EDTA, not too much, otherwise the cell will be crushed and RBCs will be breakdown, and the result will be not sufficient. During the initializing of the wind ray, I am doing the rotation of moving the sample in up and down. Now the machine is ready. As you seeing, there is an ambient temperature because the temperature is little high for the machine. For the room, more than a room temperature, I have to enter the details, which is I have to enter the only lab number because of the security issue. Now just push the button of CBC, it take the samples, and it will take around 2 minutes to count all the parameters of CBC. Firstly, it is doing the WBCs, then it is counting the IBCs, and now the counting is finished. It will give the print through the thermal roller, as I already put the thermal roller, and it can also show on the graphs. Now I have removed the centrifuge samples which I put, then take the samples, and now I am going to do the serology of these samples. The labeling is very important in the lab because every sample is unique. Now you have to take the samples in the sample place and wait until the control line is on the sample. Now you can see there are two lines if the test is positive. The first one is the test line and the other one is the control line. If the control line is shown, it means the test kit is valid and test is valid. If the control line is not shown, it means test is not valid.